webinar because it's kind of pushing some next generation stuff from using single cell sampling into a more high throughput method on Celigo. So first, I'll go give you an overview. We're going to give you a little background and introduction of what the content is and who's actually using this kind of application. Then we'll talk about the plate preparation for today's sample and why we're going to use that. And then we're going to talk about how we're going to easily image this on the Celigo and do analysis to become more of the high throughput method. And then we're going to review the data and the calculations of how do we prepare, get the sample calculations to be showing cells per mil on your output. And then we're going to give you a sample layout for unknown samples. Okay, so when we're going to go into the who is actually using this type of assay on Celigo, we want to first understand what it is that we're actually doing. We're actually monitoring the cell viability and concentration measurements for multiple suspension samples on the Celigo. So basically, if you're into cell line development where you have multiple flasks growing with different cell populations, different growth rates, CHO cells are commonly used, hybridoma cells, or if you're an immuno-oncology scientist, then you're looking at splenocytes where you're isolating them and you need to have a viability and cell concentration immediately to move on to the next step. These are the type of people that have multiple cell samples of unknown concentrations and viabilities, but they need to know them quickly. So typically people are using the old school method of the hemocytometer, something we all started working on in the lab when we first get into the lab. And then you get into the automated methods such as the Celometer where it automatically counts it, or the ViaCell. But that kind of is a low throughput where you have one sample at a time. But if you're working on, say, 100 samples, that got to get laborious over time. So what today I want to do is kind of move into the next high throughput method, which is more of a plate-based cell counting and viability measurement on the Celigo using this reagent called AOPI, which is actually acrid orange and propydium iodide. It's nothing new, it's been around forever, but we're going to use those quick staining fluorescent dyes to actually measure the viability pretty quickly on the Celigo. So, but there's some important things that you need to kind of know before we get into that sample preparation. So this is what this training session is going to be about, is to kind of educate you when you have unknown samples of different concentrations that you kind of want to understand what is the sample volume that I want to add, what are the sample concentrations in the dynamic range that I can have confidence that I'm going to be able to get accurate counts out of the Celigo cell counting. So we have to determine that your sample has to be diluted sometimes when it's super concentrated into an appropriate range that is accurate for the Celigo to detect it. So today's example or training session is going to teach you how to prepare a plate that helps educate you to find out what is the proper sample dilution and what is the sample volume that you want to add to your plate for your cell counting AOPI assay. So that's really where we're going to focus on the training session. All right, so this plate preparation, we are going to use a known sample. So basically you have some healthy cells growing in there in your flask, and what you want to do is you want to make sure you have a high viability to do this because you want to have good viability for your cell counting. You're going to measure it on your old school method, which is basically your Celometer or hemocytometer, and then you're going to get that known concentration. You're going to centrifuge that cell down to a pellet and resuspend it to a known concentration. So this would be 8 million cells per ml. That's where my starting known concentration is going to begin. Then I want to prepare 5 tubes at the following concentrations. I want to have 200 microliters of each of these 5 tubes. So you'll see label tubes 1 through 5, and you'll notice tube number 5 is the concentration I asked for. It was 8 million cells per mil, and then as you go up, you'll see it's 4 million cells, 2 million cells, 1 million cells per mil, and then 500,000 cells per mil, and that's 200 microliters each. So these are what we're calling our known sample concentrations. So next we'll want to serial dilute our AOPI stock solution. It's going to be a 20-fold dilution. So typically I'll dilute it in HBSS with some HEPIs. That's usually a good buffer for samples that might be a little bit finicky. They have a little bit more TLC in that buffer. So that's going to be your working solution of AOPI. And then next you're going to want to prepare a plate. You're going to get a Greiner 655090 plate. This is a black wall, clear bottom plate, and you're going to pipette 190 microliters into rows B and C. And you're going to skip the outer row of the entire plate, so you're just working on the inner rows. And then 100 microliters goes into rows D, E, F, and G. So this is basically just your diluted AOPI solution, and then you're going to put in your sample set. And so note, you're going to only pipette 10 microliters of the sample sets in those tubes 1 through 5, and you're going to put 10 microliters of that sample into rows B and C. So this is going to be just a very small amount that goes into the top rows B and C of that AOPI solution. And then you're going to serial dilute. You're going to take 100 microliters from row C and pipe that into row D. So that's basically a twofold dilution on that. And then you're going to continue to serially dilute those rows. So D goes into E and then F and then G. So you get all the way down to G, and then you're going to remove that last 100 microliters that's in row G, and you're going to discard it. So you have pretty much 100 microliters in all wells in the plate except for the top row, which is B. So here you have a visual representation of the instructions I just gave you. So you're going to pipette a 20-fold dilution of AOPI solution into rows B and C, and then 100 microliters of that same solution into D through G. And then for your samples, you're basically going to have 10 microliters. So here you have 2 plate map layouts. So one is showing your sample number. We have samples 1 through 5 in quadruplicates, and then this is the volume they're actually pipetting into those rows, B and C. So you can see actually you're going to have duplicates of B having sample 1, and then you're going to have a twofold dilution of C here, basically by pipetting the same amount. And then what we're going to do is basically take 100 microliters of this solution from row C. As you see the map displayed down here, we're going to pipette out 100 microliters out of C and put it into D, and then we're going to again serial dilute D into E, and then E into F, and then F into G. So this basically is your serial dilution of sample, and the last row, G, you're going to discard the 100 microliters so that they have 100 microliters in that last row, G. Okay, so here is the plate map dilution factor for your samples that you just prepared. So row B is your 10 microliter sample. It's what I would consider neat, where you have the rows below that are all serial diluted. So you have 2, 4, 8, 16, and 32 dilution factor of your sample. So we just talked about the plate preparation. Now we're going to move into how you acquire the image and analyze it on Celigo. So on the Celigo, we're going to actually pull out the plate profile for the Greiner 655090. So that's a 96 well plate. And this list is alphabetical by vendor. So you have BD Falcon, Corning, keep going down, and then it becomes numerically once you get into the manufacturer you want. These are all different plate formats: clear wall, white wall, black wall, U-bottoms. We want this particular catalog number, 655090. It's a black wall, clear bottom plate. And then we'll put in a plate ID. I've already got one here. It's Viability High Throughput AOPI. And then we'll just load the plate. The system moves over to the Scan tab. And here in the first top column on the upper left, we have to select the application in the viability section. So this is going to be a 2-channel acquisition. So we want the live plus dead application. And then on the right-hand plate map, we're going to select the button selection and then highlight the wells that we want to acquire images in today. Again, we're going to skip the outer rows of the plate, and then we'll click back on the navigation and navigate to a well that we can start setting up our image parameters. Let's set up the live channel first. So the illumination color for the live is going to be green, so we'll select. We're already on the live channel. You can see here we have two channels, live and dead. And what we'll do now is change the illumination to green because AO comes through in our green filter wavelengths. And then we just click snap to see where we are. So we have the sample, but it's a little bit out of focus. So we'll use the focus setup button and then click register auto. The system will study the green channel objects to see where they are in focus. It goes up and down in Z, similar to like what you do on a microscope. And when it's in focus, it will then register the hardware focus beams on the bottom of the plate. So that will be the focus mechanism and used for the entire plate. So you can see the cells are in focus. The ideal object pixel intensity. This is when you hover your mouse over the object of interest. The pixel is red down below at the bottom of the screen. It says, you can see I've got a little bit of background, but my object pixel intensity is about 130, which is good. That's the middle of the camera range. So the more separation you get from the object to the background, the easier it is for the software algorithms to detect the objects in the image. So this exposure is just fine. Now we'll switch over to the dead channel and set up the image acquisition parameters for that. So first then we'll change the illumination to red. This is for our PI stain. And then we can click snap just to see what we're at. I typically use snap when I'm in fluorescent imaging because I don't want to photo bleach my samples. Snap just takes a quick picture, it doesn't save it, it just displays it. And then we'll use the find focus button this time instead of the focus setup because really I just want to see where my cells are in focus, and then it will show the number above that button. It says current focus offset. It'll have so many microns negative from the green channel. That was our registration channel. So then we click set offset. That number -33 is populated as the focus offset for this red dead channel. So when I acquire the images with the green, it will move then -33 microns and take the red picture so that both channels are in focus. We're all set. The illumination looks great. These are really bright PI dead cells. We just have to click start scan. All right, so as we are over here now on the Analyze tab, there's only a couple of buttons that we actually have to select. The default settings are pretty darn close, so let's just go over that. So first we're on the live channel, and so just remember when you're changing parameters what channel you're on. If you just select this live button, it'll show you can have the ability to switch to the dead channel. So for the live, we're going to change or turn on the check separate touching objects, and the minimum area acceptance we want to have is 20 micron per pixels. So we want to eliminate small cellular debris that's not really a cell. We'll do the same thing for the dead channel. You just change the channel over to the dead, check the box for separate touching objects, and the minimum area is 20. Those are kind of just right off the bat things that I know I like to do. You can see the first well has been acquired and saved into the computer, so we can start looking at it. So here we have a display of both channels. The AO stained green cells are alive, and then the dead red cells are PI stained. The cool thing about this assay is that the AO stains all cells, and the PI will stain only the dead leaky cells, and it neutralizes or quenches the AO stain. So you can see that I only see PI stain, no crossover into the green channel. So it's purely dead cells that are PI stained. And then the AO is We have our graphic overlay, which is properly identifying the AO alive cell stain, and then we have the dead graphic overlay properly identifying the PI stain. You just turn on and off the graphic overlay. I usually do this a couple times to see if there's something that I'm not quite getting, or we're picking everything's okay. Here I have some very faint PI stain cells that I probably would consider this cellular debris. Maybe the cells died a long time ago and it's not really a current representation. So I would probably increase the intensity threshold, meaning pick up less dim cells, or not pick up those dim cells. And so moving that intensity threshold from 4 to 5, it will only pick up brighter PI stain cells and ignore the darker ones. Oh, and also on the lower left, have the check auto analyze box on, so that every time you do a parameter change on the left-hand column, it will automatically update the graphic overlay in the image display area. So now you can see that just changing that intensity threshold up a little bit, we're not picking up those faint dead cells. I think it's just cellular debris that's been left over. It's not really a true dead right now. Okay, and at this point, we're still looking at the analysis parameters. What I'd like to do now is to move over to the edge of the well. So typically this is something people don't think about looking at, but we do need to come over here on the analysis and look at the edge of the thing, the objects being picked up. So because the transition goes from a green background into black, sometimes this is detected as an object. But I'm going to help set up the parameter so it ignores these edges where the image of the well wall gets detected as an object. So first thing, these are really long objects. You can use aspect ratio to eliminate those things. That's basically when you have a circle that's perfect. It has a length versus width of 1. So anything less than 1 is a little bit more long. So as I increase this value of aspect ratio up, you can see the long objects start to disappear, because that's the threshold. I'm basically saying anything that's really long has a length versus width that is, you know, very skinny, then basically I want to eliminate it. So I'm only going for round objects. So that's an easy one. I mean, you could use the well mask and shrink it, but then you might also miss some of the live ones that are real samples. And sometimes cells aggregate over to the edge of the well. So I sometimes use the aspect ratio as a first parameter if I know that I have a clean edge. So just moving the well around a little bit, you can kind of see some behaviors of things that you may or may not want to be detected, and that helps you set up the parameters that you're going to use for the entire plate. So I'm just moving the plate or the well around, and then I'm upping the aspect ratio. Probably you could go up to maybe 0.4. I'm just using this on a 100th micron up. What do I say, 10 micron up increments to show you how little increments can make a big difference. There are some small objects up here that are really not cells either. So let's change the minimum area requirement. We'll go up to 30. That looks pretty good. So this is really cell type dependent. These are Jurkat cells that are pretty tiny. CHO-S cells have a bigger diameter. You might want to move that minimum area up to 50. So it really is cell type dependent. But once you find your settings that you like, if you look at the top portion of the screen, there's a little save icon. So you can save these analysis parameters for your cell type and not have to go through this every time. Just figure it out once, and then you got it saved, and you can recall it next time you do your same assay. So once you have those analysis parameters set up, then you just hit Start Analysis, and it'll apply those parameters to all of the wells that you are acquiring images. So you'll notice the progress bars down below at the bottom of the screen are both acquiring images and analyzing at the same time. So that's the nice thing about Celigo. You basically can do things simultaneously. You don't have to wait to the end. But while we're waiting, let me describe some of the things that you see on the screen. The data output measurements on the left-hand side, these are the calculations of how you actually get that. The live count is actually the live cells in AO divided by the total multiplied by 100. And then so forth for the dead, you get dead count divided by 100. The live corrected is basically live minus double positively stained cells times 100. So that's basically if you have cells that are slightly dead, they're just starting to get leaky, then taking in the PI stain, we're going to remove that. That's what we call live corrected. So it's pretty much the live only. And so down below you'll see not displayed values. It says live corrected counts. Basically you're going to have the live minus live plus dead, and then the total counts, which is live corrected counts plus the dead counts. That's how the total is actually being calculated in these data measurements. So let's just go ahead and save this experiment. So if you're going to run another plate right after this or another day, you've got all these parameters set up perfectly for your assay for AOPI. You can save this experiment and it remembers everything, the wells that you want to acquire, the illumination, the application, the channels, the exposures, the analysis settings. It saves it all so that you can just pull that up on the very first page and it basically loads up all the parameters and you just hit start scan, start analyze, and it goes through everything automatically. That's the nice thing about having the save button. You only have to remember it once and then recall it for the next couple of plates. All right, so looks like both analysis and acquisition are finished. And let's take a peek at the data. So here we're looking at our lowest sample concentration, which was 500,000 cells per mil. And if you scroll in on the image, what we want to see is how are the objects being detected? Are we liking what we see? See on the left-hand column, it displays the data for this particular well. We've got 90% live cells, and we don't have many double counted, so that's good. So our live corrected and live are very similar. So I think that, to me, says that we have a really healthy set of cell samples. If you have some double counting, that means you might have some cells that are on the way out and dying. And now we're just looking to turn on and off the graphic overlay to see that we're properly detecting the cells. It looks really good. In the lower densities, it's looking really good. And here are dead cells. So you can tell when you have a lower density that the cells are spaced out far enough from each other that you can get the proper identification of whether it's alive or dead. But we'll notice when we go up to the higher well densities, when you have too many cells in the well, you might have problems where you get clumpiness. The cells haven't quite separated from themselves, and you're going to get double counts or even excluded counts because it's not a round object anymore. Here we're in the middle cell density. We're just going to continue to kind of observe how is the graphic overlay detecting the cells. It's looking pretty good. I see it's doing a good job. Very little is not picked up. So I would say this is pretty good. I visually have confidence this is the right count. And you can confirm you've got the same percent live cells, about 90%. And let's move up to the higher cell densities. And now you'll start to see where you have cell clumping. And those cell clumps don't get detected because we use the aspect ratio. We could try and tweak the settings a little bit, but I think it would be easier just more for this exercise to understand what cell density and dilution do you have confidence in the range of your cells. This is the highest concentration we plated. It was supposed to be 10 microliters of the 8 million cells per mil, and then we have a twofold dilution into this row C. So this allows you to see when you have a higher concentrated sample, just by diluting it out, you start to see where you can have a little bit more confidence that your cell counting is accurate because it's a little bit more diluted. And it's looking a little bit better, more like the lower density cells that we saw in columns 2 and 3. So I would probably say at these high cell concentrations, you probably want to dilute down to, what is this, about 4, dilution factor of 4. That looks really good all the way out to the edge of the well. Now, if you go out to the edge of the well on the higher concentrations, you're going to see where cells tend to clump a lot is on the edges. As soon as the image displays, there it is. So you can see it just starts to get a little bit ugly. There's nothing you can do about it. I think your better bet is just to dilute your sample more to have confidence on those higher samples, higher concentrated samples. Even the duplicate, so you've got 50,000 versus 60,000, you're kind of wondering, sample replicate-wise, it's not very accurate. So let's export this well-level data into a CSV file and open it up in Excel. And so I'm going to show you next how do you re-back calculate it into a cells per mil concentration. And I want to show you also with our dilution factor where you can start to see you can have confidence in the cell dilution factors. Okay, so we just covered imaging and analysis, and now we're going to go over the calculations and data from that plate. So these are of known samples, so you kind of understand when you put in a known sample, it teaches you how to prepare your unknown samples. So here's our exported file, and you see the first set of information is your plate information, and then we go into measurements, and there we have the % live, and again here are the outputs and how they're calculated on the software, and then you have the live corrected, so this is truly your live very happy cells, and then you have your live counts, dead counts, live plus dead counts. And then your total is not displayed, but it's using the live corrected counts plus the dead counts. The live plus dead count is truly your double counted, double positive stain cells. And I don't have that many going on in this sample set. But for me, again, I feel like if you get a lot of cells in this area, you're probably getting an unhealthy cell line. It might be at the point where your cells were stained with AO but are quickly dying with PI. So that's just an observation I've seen. Okay, so let's take this information and move it into calculating how our cells were diluted. All right, so here on the second tab I have a calculation sheet. And let's go up to the top and I'll show you the information I'm using. So here I'm using the live count, which is the corrected. It's only the live minus the double positive stained cells. So it's really truly the only live cells. All right. And I'm just bringing those wells over from the previous tab. And then we have the dead count here. The replicates look really good. And here I'm using those two to give me my calculated total count. So it's the live count corrected plus the dead count is now giving me my total. So this is the value that I'm showing you how I'm calculating the total. If you would prefer to do your cell concentration on the live only, live corrected count, you can do it that way too, but it depends on what you're going for. So here on the right hand side of the total I have provided some columns showing the theoretical of what I intentionally wanted versus the actual of what was calculated from the Celigo from the plate. I want to see how accurate, and I'm using an average of the two wells to show how well did I get to the value that I thought I was plating with the known samples. So you can see the lowest cell sample we had was 500,000 cells. I was expecting around 5,000 cells in the well because I only pipetted 10 microliters in. All right, and so then we have done that same math all across the plate. And if you plot the theoretical versus the actual, you can get this correlation plot. And when you plot that, you can get an R-squared value to understand how confident you can be that your sample dilution was properly counted. So you can see that the lowest concentration of 500,000, I've got 0.9907 R-squared value, which is very good confidence. But as you scroll down and see the other sample sets, the sample set that's in the middle, the gray one, I've got the highest confidence in. At the lower end and the very high end, it's still good, but you can see where it starts to drop down a little bit, but it's still good. So this will help you determine what sample set range and dilution you want to pipette into your plate. So let's use this information and back calculate to get the cell concentration and the cells per mil value, which people typically report when they're working with suspension cell populations. So here I'm using the formula up at the top. You can see I've got the dilution factor times the cell count divided by the volume that I actually pipetted into the sample times 1,000 to bring us back into the milliliters instead of microliters. That will give you your cell concentration in a cells per mil formula. This dilution factor plate layout is what I showed you earlier in the plate layout. It will help you back calculate your sample set. So that's our lowest sample size, which is 500,000 cells. We expected 500,000, and if we take an average of that entire set, once they're corrected for their sample dilution, you can see that it's pretty darn close to 500,000. It's just a little bit above that. And if we apply that same formula to the rest of the samples, sample set 1 through 5, you'll see that I have my expected here and my actual average, and I'm plotting that, and I'm getting a very tight R-squared value. And also something you can utilize is the CV. So if you have a large variability in that sample set, basically that might give a little concern that maybe that's the lower end of the sample is too dilute. Maybe we shouldn't use something of that sample dilution factor for that sample size. So once you eliminate that, you can see how the CV goes up for that lower end. So it might be too dilute for those lower concentrations. So this is basically an exercise to help you understand when you have so many unknown samples. This one helps you understand the dynamic range of what confidence you can have in the cell counting for the plate-based assays. So at this low density, you might want to eliminate samples that come out with 100 cells per well, because you just may not have the highest accuracy when you have diluted it out that far on the low samples. Here on the upper end, when you're looking at 8 million cells per mil, when you do this in a sample serial dilution, you might have too many cells in a well. You saw in the image how we're not picking up the clumpiness. There's just too much clumpiness in there to detect accurately. So you might not want to have those samples in that high concentration in the well. So you can see wells that have counts up to 50,000 cells per well in a Celigo image. You might say that's too high on the end of the range, so let's not count those in our cell dilution. If we see values like that, we're not going to have confidence. So we want to have sample sets that are above 100 cells per well, but below, say, 30,000 or 40,000 cells per well. So looking at this exercise, you want to kind of figure out, well, what would be a good dilution factor for the entire range of samples that I might have, something that's really dilute and something that might be really high concentrate or a lot of cells per mil. So I would probably pick something in the middle of this whole exercise, something like 5 as a dilution factor. So you can use that with confidence because it seemed to have a good dilution for the whole dynamic range of the cells that were covered in our exercise of known sample sets. So this is what we've just gone over is the calculation and evaluation of the data. Now I want to show you a sample layout for unknown samples. So we just went over an exercise for known samples. Now we're going to go show you how you would lay out for unknown samples. So you're going to prepare for this example exercise a dilution factor of 100 from your stock cultures that you're growing. How do we do that? Well, we're going to actually use 2 plates today. So this is going to be a dilution plate and then an imaging plate. So first step, we're going to prepare the 20-fold diluted AOPI solution like we did in the first experiment. And then we're going to prepare 2 plates. We're going to pipette 90 microliters of this AOPI solution into what I call a pre-dilution plate. It's typically, I find a U-bottom plate is most functional for this pre-dilution plate. And the second one is our imaging plate, which we use today, which is the Greiner 655090 plate. That'll be our imaging plate. You're going to put 90 microliters of the AOPI solution into that imaging plate. Then you're going to pipette your samples, your unknowns. You're going to just do 10 microliters into the pre-dilution plate. So you have 10 microliters of sample 1 goes into well A1. And then you can see the numbers of the unknown samples in that pre-dilution plate. So this is just a 10-fold dilution in the pre-dilution plate. Then you're going to take 10 microliters out of this pre-dilution plate and pipette it into the imaging plate below. And we're going to do that in triplicates. So you can see sample 1 is pipetted 3 times in wells A1, A2, and A3. So you get a triplicate. After you've pipetted all your samples, I prefer to centrifuge the plate down if you want to image it quickly, or you can wait 10 minutes to let the cell sample settle down onto the bottom of the plate. I find that when you have 100 microliters in the well, it settles the cells down pretty evenly. If you have higher volumes, it will probably aggregate to the edge of the well. So 200 microliters, a little high for settling, but 100 microliters seems to settle evenly. So that's pretty much it. We've covered how and who is actually using this type of assay in the world of cell line development or in immuno-oncology. We showed you how to prepare known samples and to understand the dynamic range of your samples, how to prepare that plate. I showed you easy setup for image acquisition and analysis on Celigo and how to save that experiment for your next plate that you want to run, and then how to calculate to get your cells per mil. And then I showed you an example layout of how you would actually run unknown samples. So that's a lot. I know that was a lot, but it was a really good experiment and assay to kind of teach people how to do cell suspension samples of unknown and known populations. So at this point, I'd like to open up the forum for some questions that I might be able to answer.
Thank you, Sarah. That was a wonderful presentation. As Sarah said, we are opening up this time to answer any questions our attendees have for us today. So if anyone has any questions, please type them into the questions bar, and I will receive the questions and read them out to Sarah for a Q&A session. I do see that we already had some questions submitted throughout the webinar, so Sarah, I'm going to begin the Q&A session.
Okay, great.
All right. So question number 1 asks, is AOPI commercially available combined, or should it be prepared by mixing individual components? And is there a staining protocol available?
It's commercially available from us. We actually sell it at the Nexilum website. If you go to our website, you'll see that we sell AOPI already prepared in the correct concentration for you, and then you can follow this protocol. If you want the actual protocol that I use today, just email us and we'll get that to you.
Great. Okay, so we have another question that's come through. It states, what settings help get pixel intensity between 125 and 150? Is it exposure time or gain, or can you elaborate?
Yes, absolutely. Very good question. So pixel intensity, so that's basically when you expose the illumination to the sample, it fluoresces according to the dye that you've put in there. The more exposure you have, the more the pixel intensity will go up. As you can see, the AO sample had a lot of background, so if you turn up the exposure, it might also bring up the background, so that's kind of a balancing act. I chose 10,000 microseconds because our sample had pixel intensity of the cell object in the right camera range, which our range in the camera is from 0, which is black, to 255, which is completely the maximum. So I'm shooting for a value that's in the middle, which is like 130, 150, something like that. When your exposures get too high, say today I was using 10,000 microseconds, but if you get up to 100,000 microseconds, the scan might take for a long time if you're going to do a full 96 well plate. So rather than turn up the exposure anymore, I like to move the gain up in groups of 100. So I never really use the gain unless I absolutely need it. The exposure is the first tier that I use. When I get up too high of an exposure, I start to use the gain that allows me to bring down the exposure time to shorten the scan. And again, if you start to see too much background, you're going to want to dilute out your fluorescent dye or maybe do a wash to remove the background. As you noted, the protocol I described today, I had diluted the AOPI stock solution 20 times. Basically, that brought me down to a low enough background. I probably could have gone down lower if I wanted to, but I chose the 20-fold dilution for our stock dilution, and that basically gave me good signal with low enough background. It was around 70 in the background. I would prefer to have it lower, but I was able to detect the cells with the default analysis settings. That's kind of the other thing that I shoot for. When I'm trying out new dyes for the first time, I'll do a cell or a dye titration to figure out what is the best concentration for me or for my sample to have where I don't have to play around too much with the default settings that are displayed. Typically for fluorescence, the default settings are 4 intensity threshold and 10 diameter. And so if I can get my dye or sample to be properly detected with those default settings, then I have to spend less time on the analysis tab.
Great, thank you, Sarah.
Mm-hmm.
All right, so we do have another question. It states, what is the difference between the two save icons on the bottom left of the panel while saving experiments? Okay,
so one is save and the other is save as. And you guys know that you have those same icons in other Windows applications. Save as is if you've never saved it before, so it's the first time you've ever saved it. You just give it a new name. And then say you've saved it with that new name, and then you go to the Celigo again, like in that current session, and you make a couple of changes. Maybe you change the intensity threshold or select a different set of wells, but you're still in that current session, but you want these new changes to be included. So then you use the other icon on the left, the smaller, narrower one. That's the save. Basically, it's adding those new parameters or new changes to that currently saved session or saved experiment that you just saved.
Great. Another question?
Wait, before we go on to that, let me just point out that as soon as you close that session, meaning go back to the Start tab, basically that save experiment is not editable. You'd have to reload the saved experiment, and then you can recall it, and it should update with any new changes. So to make changes, you have to be in the Celigo software with that saved experiment loaded to make new changes if you've basically gone out and come back in.
Thank you for clarifying. Okay. Another question is, will this analysis work for non-suspension cells, adherent cells, non-rounded cells after drug treatment?
For adherent cells, those typically are a little bit more challenging because they're not that round little aspect ratio that we were talking about. And the staining for the AO might be a little bit dimmer because as you have a flattened out cell, the pixel intensity might get a little bit closer to the background level. So I can't say that I have done AOPI on adherent cells. That's kind of sad. I would prefer, honestly, to use Hookston PI for adherent cells. That's because Hookston is the nuclear stain. AOPI gets a little dim and fuzzed out when you have adherent cells, so I would prefer for a viability assay to use Hookston PI for adherent cells.
Thanks, Sarah. Okay, I'm not seeing any other questions come through, so I'm going to give you guys just a couple more seconds to see if we get any other questions for Sarah. While we're waiting to see if any other questions come through, I just want to remind our attendees that we do encourage you guys to submit any topics for future webinars. We would be happy to hear any suggestions you have. If there's anything else about the Celigo you'd be interested in learning about, or any of our other instruments and how to use them, feel free to email us at support@nexcelum.com with your suggestions, or there is a short survey you can take at the end of this webinar that's about 2 or 3 questions long, and you can type in your topic suggestions there as well. Okay, so I'm still not seeing any questions come through, so I guess that will conclude our webinar for today. Thank you for all of our attendees for joining us today, and Sarah, thank you for a wonderful presentation. The session today was recorded, so everyone who is registered for the webinar will receive a link to that recording, and you can watch this webinar again or pass it along to a friend or colleague if you wish to do so. Thank you everyone for joining, and I hope everyone has a wonderful day.
Thanks, Amy. Thanks, everybody. Talk to you later.
Bye.
Bye.